Muhammad Umar Farooq Biological video. Please make sure to subscribe, like, and share. Ask the question for answer and comment about the artwork. Today we will talk about the Chlamydomonas. That Chlamydomonas is the we will talk about the kingdom Plantae and Chlorophyta. There's a division Chlorophyta and class is Chlorophyceae and order is what vocals. Family Chlamydomonaceae and genus is Chlamydomonas. So here is that the kingdom Plantae. That's related to the kingdom plant, which that is autotroph, and that is using the energy from the light to produce ATP and NADPH by the Calvin cycle and electron transport chain through light. So here is the basic general structure of the Chlamydomonas. That is the unicellular, contain flagella. That is the pair of flagella, which that is the anterior part of the cell. So here is the eyes part, which that is using for the detection of light, and there's a contractile vacuole which is important for the absorption of water and releasing of water. So here is the chloroplast and cell wall is present. The chloroplast is used for the, that is cup-shaped chloroplast is present, and cell wall is made up of peptidoglycan and cellulose, and cell membrane is the made up of phospholipid, and pyranide is used for the storage of food. So here is we will draw a cross section of the Chlamydomonas from here. That is the stellate chloroplast is present, is looking here from the upper side of the cutting you can see in the figure. So the morphology of the we will discuss about the morphology. Here is the microscope, with that is the 1500 time more resolution as compared to the human eye, which show that the structure is like this. And if we will talk about the electron microscope, which that is the analysis is the 1000 time more resolution as compared to the compound microscope, and there's the resolution is show the whole study of the Chlamydomonas cell. So that is the unicellular, and that's the eukaryotes because the true nucleus is present. And here we will talk about the flagella. There's a flagellum is contain plasma membrane, and there's a plasma membrane covered inside is the centriole. That's the inside. So here you can see the figure. So next we will talk about the whole structure, which that is shown by the study of electron microscope. That is the important, that the filament, which that is the flagellum, which that is embedded in with the plasma outer membrane, outer and inner membrane, which that compose of phospholipids, and there is the different types. Hook filament junction and hook are present is here, and L ring further is embedded in the peptidoglycan layer, and that P ring is further divided into MS ring. So that is the inner membrane is present is here. And type 3 basically type 3 secretion system that is present inside, and that is the motor complex is shown in the figure. That is the morphology of the flagellum attachment. So next we will talk about the reproduction. That is the asexual reproduction is first of all we will discuss about here. That is the flagellum disappear, protoplast shrink, shrinkage of protoplast, and organelle disappear. There's a disappearing of organelle, that's becoming a whole structure is without flagellum, and there's the specialization of organelle will be disappear, and there's the structure will be occur in basically so I will explain with a diagram. So here is the flagellum that is disappearing, and that's begin to create a line for the cleavage. That is the cleave a line which that is the nucleus, for example, is present is here, that is nucleus is divided in the further is transverse way, and that's convert into double into triple and sorry tet four time division, that is the four cell will be produced, and there's the further cell division by the asexual reproduction to produce a too many cells, which that is too many cell is a Chlamydomonas, that is here is showing in the figure. So in this type of reproduction, that is the multiple Chlamydomonas will be released by the um, rupturing of the plasma membrane or hardening of the outer layer is by the deposition of cell wall, that is conversion of cytoplasm, that's the cytoplasm decrease and plasma membrane will be decreased, and that's the flagella will be disappear. Here is I can mention about that things. So here the flagella is disappearing and protoplasm shrinkage organelle disappearing, and that's why is the favorable condition will be preferred to and divide the cell, and that's division of the cell will be give a multiple number of the Chlamydomonas, which that is released in the fresh water, and sometime in the pond and ditch is also uh, exist the Chlamydomonas, and there that is the freshwater alga, that is the present in the freshwater. So that is why it is known as freshwater algae. So that's the freshwater algae is unicellular and eukaryote because it's a true nucleus is present in the Chlamydomonas. So that's all is shown in the figure. Now next we will talk about that is the sexual reproduction. So the sexual reproduction we will understand first of all. The reproduction is asexual vase looking here, that is the one cell which that is going into the two cells and multiple cells, that's conversion of the releasing by the rupturing of the Chlamydomonas, rupturing of the wall to release the Chlamydomonas, and that Chlamydomonas is known as asexual reproduction by cell division. On the other hand, we will discuss about in here that is the mother cell gametes, that is the liberated gametes which that is produced by the meiosis. So that is the result of the meiosis, that the female and male gametes will be produced. There's a male and female gametes is isogamous because both are equal to each other and not differentiate to each other, that is a female and that is a male. So that is why it's fusion to produce again for example there is the four flagella by the fusion, and that four flagella is come from here, that is known as quadriflagellate. Quadriflagellate is known as is here. That's a quadri mean four flagella is present, and basically so that is why it is known as quadriflagellate. That is the zoospore because it can be moved into the water. So the zoo mean animal, so animal mean it can move from one place to another. So that is why here is the four cells, and that's releasing of the releasing mechanism, that is the after the meiosis it can be again produce Chlamydomonas, and that is the 1n, and here is the 1n asexual reproduction, and sexual reproduction result is the 2n, and again will be 1n, the haploid to diploid. So here is the sexual mean So in the presence of sexual, it's haploid, and in the presence of asexual reproduction is known as diploid. So here is we will talk about the practical. So here the fresh water which that is contain um, algae, and that's algae drop by the slide, and the slide put into the microscope and analysis. So here we will talk about the analysis of their movement and their study of the cell structure. So here the Chlamydomonas, which that is the moving into the water. So first of all, we will talk about the phototactic movement. The phototactic movement is toward the photon, there's a light, and there's a positive phototactic movement is toward the um, light, and there's a flagella will move backward. There's a whiplash structure of flagella which that is move backward, and the Chlamydomonas will move forward. So here the negative phototactic movement is meaning the away from the light. Now here I will discuss about the structure of the cell of Chlamydomonas. So here is the flagella, and here is the outer layer is known as cell wall, and inner layer is the plasma membrane. And that's here is the Golgi apparatus, and mitochondria is the specialized organelle, and the anterior portion contain the nucleus, and the pyronoids are present which that is store the some food. And here is the starch granule, which that is the starch granule are present into the chloroplast, and that chloroplast contain filament, and there's filament contain thylakoid and thylakoid membrane which that contain plastoglobule, and this plastoglobule is a very important function. And here's the basal body, and eye spot is here black color in showing in the figure. That's black color is eye spot which used to phototactic movement, and that's phototactic movement is important to detect the light for photosynthesis, and photosynthesis is the necessary for the autotrophic lifestyle. Here is plastoglobule. That's plastoglobule is very important, which that is the thylakoid membrane, which outer projection of the thylakoid membrane which covered by the protein, and that's using basically here we will talk in the next video about their process. But here is the plasma membrane, thylakoid membrane, and that's covered by the outer projection is here is the protein which that is bind by the so here is the PG uh, core, which that is PG core, and here is protein binding, and here is the lumen and ATP synthase enzyme which that is attached by the and different channels is attached for the light reaction. So that is the overall of the um, diagram of the Chlamydomonas. That is green color shows that the filament of chloroplast. So it's the cell wall which that is made up of peptidoglycan and cellulose. The cellulose is the basically alpha 1 to 4 linkage of the sugar, that the same sugar is known as homopolysaccharide. That's a homopolysaccharide is made up of same sugar molecules arrangement in the same way. So I hope you make sense about that diagram and understand about the Chlamydomonas, that is the type of unicellular algae. So thanks for watching.